like to invite Ana Maria Torres to talk about the work on genomic-assisted breeding in faba bean, please. Thank you.
Thank you very much. I'm glad to present you the genomic-assisted breeding in this crop in faba bean. Well, faba bean is one of the oldest crops grown by men and since the beginning of the agriculture. And due to the high protein content and the high yield potential, faba bean is today the fourth most widely grown cold-season legume. So the cultivated area is nearly 2.5 million of hectares, and the world production of 5 million hectares, being the main producers China, Ethiopia, Australia, and France. Faba bean is a partially allogous crop with a chromosome number very small, but only 6, but very, very large chromosomes that are easily observable. And for this reason, faba bean was one of the modern cytogenetic plants in the last century. No wild relatives are known, and all the crosses that have been tried with relative species have been unsuccessful. Nevertheless, there is a great variability between the domesticated pool, which is at the main center located in the Mediterranean region, with secondary centers in the Nile Valley, in South America, and the Central and East of Asia. Originated in the Middle East, the long history of cultivation, the wide distribution around the world, and the mating systems, and the response to the human selection has led to faba bean to be one of the most variable crops species, with a wide spectrum of color flower and also seed shape, color, and size, as well as many other agronomic characteristics. After the selection, we can distinguish 4 botanical groups, the major types in the right side, which are the mainly used in the Mediterranean countries in green, the Quina and minor types, which are mainly used for the animal consumption, and Paucijuga types that are thought to be very close to the wild progenitor and are very small, round, black seeded types that are mostly cultivated in India. As in many other crops, the faba bean breeding problems, the main ones are the resistance to biotic and abiotic stresses, and also the increase of yield, and to find appropriate phenology to be adapted to different environments, all of them controlled by quantitative trait loci. Then other problems are other interests are to enhance the seed quality, especially to eliminate anti-nutritional compounds such as low tannin and disincolizine, and also there have been some efforts to look for some genes controlling plant growth habit. All of these that I mentioned are monogenic traits. Here I go a little bit more deeply, showing you the main biotic and abiotic stresses in this crop. In our area, in the Mediterranean basin and Northern Africa, the main problem is the parasitic weed, the broom ray, that produce severe losses of yield, and also among the fungal diseases as coccidia, rust, chocolate spot, etc. Abiotic stresses, the cold and frost tolerance, heat and drought tolerance, and salinity. And in insects, the main ones that have been also studied are the aphids and the blue heat resistance. As David mentioned before, due to a great amount of transposable elements, faba bean has a very giant genome size. One of the biggest among all the legume crops is 13 GWAS, which is 3 times bigger than pea, 10 times bigger than soybean, and 30 times bigger than the modern Medicago tuncatula. Obviously, this has enormously difficult the genomic studies in this crop, also the genome assembly and map-based cloning. The molecular breeding approaches and the genomic tools used in faba bean have been parallel to the advances in the development of molecular markers and techniques. Of course, at the beginning, before the 20s, there were maps with very low resolution density and very low density, mainly made with anonymous markers such as NFLP, rapids, microsatellites, etc. Later on, the use of the comparative genomics, especially the synteny with Medicago and other legume crops, allow the use of orthologous markers and the development of sequence-based markers that were saturating the genetic maps. And finally, the cheap sequenced allow the development of RNA-seq datasets and the identification of candidate genes that have been also included in the maps using SNP markers. Among the genomic resources, I just would like to mention that as in many other crops, the genetics maps are developed selecting the contrastive parentals to develop F2 and reals, and then to identify QTLs in these maps. These QTLs sometimes have been validated in different environments and genetic backgrounds, and more recently, the QTLs intervals are being saturated using positional and functional markers with the final aim to identify diagnostic markers or candidate genes or markers useful for marker-assisted selection. Here, I just would like to mention or to present you the most relevant maps that were performed in this period. For example, these ones that I highlight here were the ones that were used at the beginning to assign the first group linkage groups to chromosome using progenies derived from trisomic plants. The next ones, for example, were used for identification of the first QTLs for Ascochyta or broom rape or frost resistant, that some of them have been validated in different seasons and pedigrees. Also, life cycle, plant architecture, or yield components and reproductive traits have been studied in Faba, and several QTLs have shown to be stable across years. So, in subsequent studies, these QTLs have been validated, and also the target QTL regions have been saturated with more solid, more efficient markers. These ones maps I would like just to highlight because this one was the first gene-based genetic map anchored with orthologous markers from the Chicago, and this one the first exclusively SNP-based genetic map. The advances in the development of sequences derived from transcriptome and the markers dataset has increased also the density and utility of the gene-based genetic maps. Here I show you the first consensus map that was developed using 3 recombinant thread lines and that show the positions of stable QTLs controlling Ascochyta, Orobanche resistance, and flowering and reproductive traits. A remarkable advance was reported by Webb et al. In 2016 that developed a SNP-based consensus map of 6 populations after mining and validating 18 SNPs from transcriptomes and also including in this map some SNPs from previous studies. This effort was very useful because it was possible to develop a Casper genotyping technology that is being used for the Faba community today to perform an accurate biallelic discrimination of SNPs and indels. And the last consensus map has been just recently published by Carrillo Perdomo in 2020 and include much more SNP markers from 3 FC populations. In addition to that, the authors also developed a transcriptome for 4 accessions that have developed an exhaustive gene-based set of SNPs that is being used or will be very useful for the identification of candidate genes of agronomic interest in this crop. And finally, I just would like to mention here the other relevant maps. For example, these that also identify QTLs for drought resistance. This is the only one reported for RAS resistance, and the last ones that was for potentials and also to saturate the region for flowering time. As you have seen, all of them are the parentals populations that are very easy to construct and represent and a powerful tool for QTL detection. Nevertheless, the QTL number and the resolution is very low since we have limited recombination, only 2 alleles, and only the variability existing between two parental lines. So, in the last years, there have been also great interest interest, to develop multi-parent advanced generation intercrops populations, magic populations. For example, this one in 2015, using 11 winter fava beans to identify regions for frost adaptation. This second one in 2018 for morphological and biochemical traits. And the last one, which is quite important because it includes 8 parentals and a great variability for both biotic and biotic stresses that is being developed at ICARDA by Fuad Malouf. Of course, the use of a wider genetic diversity of multiple parents and the combination over several generations has produced populations with higher resolution mapping as compared with the biparental mapping populations. I just would like also to highlight the importance of the next generation platform, especially the RNA-seq technology, to enhance the speed of the Faba gene discovery. Up to now, there have been reported 17 transcriptomes in Faba from different varieties, different tissues, different developmental stages, etc., that have been very useful to develop new markers, EST or SNPs markers, and also to identify genes that were differentially expressed in the interaction with ascocyta or in case of the stress, vernalization, salinity, etc. So this effort has produced exhaustive reference gene set that is very useful for differential gene expression analysis and genome annotation. In the next few slides, I'm going to show you the 4 only cases of gene discovery in Faba bean that have been reported so far. The first one was the terminal inflorescence, the terminal growth, a trait that facilitates crop management and mechanical harvesting. Using a translational genomic approach, the authors proved that the terminal flower, the TFL1, controls the determinants in Faba bean, as happens with Medicago, Arabidopsis, and many other species. So after the sequence alignment of contrasted genotypes, the authors were able to identify a non-anonymous amino acid change that allows to the development of a DCAP, which is the first diagnostic markers that was reported in Faba bean to perform marker selection of the determinate growth habit. The second example is include the efforts to try to eliminate the anti-nutritional compounds such as tannin content and the syncombicin. Tannin content is controlled by 2 complementary and recessive genes that are located in chromosome 2 and 3, while the syncombicin is located in chromosome 1. These compounds lower the protein digestibility and the energy content when they are used for animal feeding. And in the second case, the syncombicin also produces fabicin, a type of an hemolytic anemia that is produced in a genetically predisposed human. So in this case, for quality trace, the initial studies used the bulk segregate analysis in order to identify markers that were present in one segregated bulk compared with the second one. Using this approach, there were possible to identify markers that were linked to serotonin that produce the white flower in Faba bean or low syncombicin. These markers were transformed into scars that were useful for marker acid selection only when were used in the same genetic backgrounds. But until this moment, no candidate genes were identified. Much later on, using a comparative mapping approach with Medicago, Webb et al. Were able to identify the PTG1 and transcription factor that is determining the white flower in Faba bean. This CTT1 was later characterized, and another specific diagnostic marker was developed that allow now to differentiate the CTT1 from the CTT2 genotypes and also from the wild types Faba beans. Another example, the second example of gene discovery is the CTT2, recently reported using a combination of comparative genomics, candidate gene approach, and the linkage mapping of 3 populations. Using this combined approach, the authors fine mapped the ZT2 and identified the TTA as a BHLH transcription factor that is underlying this ZT2 gene that produce also white flower in Faba. This result was later also confirmed by gene expression, as you can see here in the figure. Finally, Bicin has been the most difficult Faba trait to discover, and despite there were many scars and Caspar markers developed, and also many publications related with this gene. But the position of these genes in the map still had very large intervals, and the biosynthetic pathway was unknown because this compound is not present in modern species or related species. So these facts hampered the saturation with functional markers. Just recently, your daughter in 2020 was able, using a combined approach, to identify the first enzyme that is associated with the Bicin biosynthesis pathway, which is a defunctional riboflavin biosynthesis protein. So this slide is just to summarize how was the situation of our in genomics before we started with the EUCLEG. As I mentioned before, only 4 genes for quality traits, for anti-nutritional traits, such as amines and Bicin and determinate growth, have been identified. Markers were available for marker-assisted selection. Several QTLs for biotic and abiotic stresses have been reported. Not all of them were validated, and saturation of the genomic regions are still needed to recover a reliable marker-trait association. As a result, no responsible genes are identified, and no markers are available for marker-assisted selection. As I mentioned before, Faba doesn't have a reference genome still, but the symptomy with Medicago truncatula has allowed to identify candidate genes in colonial regions, and the transcriptomes have allowed also the development of high-density gene-based maps, as well as identification of genes differentially expressed and candidates for genomic-assisted breeding. In this scenario, Statistical has been mentioned before, is based in 4 axes: crop diversification, crop productivity, yield stability, and protein quality. Concerning Faba, which is my aspect, the objectives, as for the other crops, are broaden the genetic base of the crop and analyze the genetic diversity, analyze also the genetic architecture of key breeding traits using GWAS, evaluate the benefits brought by genomic selection to create new varieties. Up to this moment, only 2 GWAS has been reported in Faba. The first one to identify frost tolerance, and the second one to identify the resistance to Ascocanthaphaba. In both cases, use the same Faba population with 188 winter Faba and a number of SNPs of 165 in the first case and nearly 2,000 in the second case. The EUCLEG represents a step forward the Faba GWAS analysis, because in this project we are analyzing 400 Faba lines for many key agronomic traits that have been analyzed in 2 environments. In addition to this, the genotyping has been performed with a Faba axiom array that has been launched by Dr. O'Sullivan from the University of Reading. Dr. O'Sullivan collected the transcriptome datasets from 5 different transcriptomes from different varieties, tissues, etc., and produced a validated SNP dataset in order to design the high density array from Affimetics that has more than 50,000 markers. This is the array that has been used to genotype the materials that we have been analyzing in our EUCLEG project. The 400 Faba lines, as Isabel mentioned before, were first multiplied for 2 years, 1 year before the start of the project, in order to keep the homozygosis as much as possible and avoid the cross-pollination with insect, this multiplication was performed under insect-proof cages. Later, we send these materials to the different partners for the different activities. We have set up field trials in Finland, Belgium, and Serbia, in which have been performed genotyping and biomen interactions with 100 accessions. Then the 400 accessions have been sent or analyzed in Sevilla, in Spain, and in Serbia to be used for GWAS analysis. In addition to that, there was a drug-resistant study performed in Germany, and the quality traits was centralized in our center here in Spain of the 400 accessions for 2 years. Here I show you summarized the 43 agronomic and adaptive traits that we have evaluated related to architecture, morphology, phenology, yield, as well as stress resistant in natural the partners score the natural occurring pests and diseases, and also, if possible, the logic tolerance, shattering, and drought resistance. And concerning quality traits, we have analyzed the protein content of these accessions, the tannin content, and the lysine condition. Well, once we had the information from the phenotypic field assays and also the genotypic information, we were able to start our first GWAS studies. And as you know, GWAS is based in 3 pillars: to have a high-density SNP catalog, to know the population structure, and to have phenotypic data points from the population you are analyzing. So by doing GWAS, you are able to identify genes and criteria associated with the traits. We have followed this scheme. After the phenotypic and the genotyping, we have analyzed the population structure and bloodness, and we have assigned different statistical models in order to identify some associations. We are also validating them with the final aim to identify the responsible genes and to characterize the genes that are responsible of a specific key trait. Once we receive the phenotypic data from the partners, we filter the outliers. We studied the broad sense heritability, we estimated this broad sense heritability, and also used the lab to adjust the data across locations and years. The genotyping data were also subjected to a quality and control analysis by removing markers with amino allele frequencies of 0.05 and eliminating monomorphic markers, of course. And the present score matrix include 352 samples with nearly 23,000 SNPs. And the missing values were inputted by allelic frequencies. As I mentioned before, due to the huge genome size, we lack at present of genome sequence and physical maps. So we make use of the genome seeker that exploits the synteny with medical concatenation to identify order and structure the chromosome sequences in the chromosomes, and also the positions of 3 genetic maps of three derived from 3 populations that were genotyped using this array. By doing so, we were able to assign to the 6 chromosomes of Vicia Faba, we were able to assign 17% of these markers, and the remaining markers were joined in a hypothetical chromosome 7. Next, we perform a population structure that, as you know, calculate the blackness correlation among individuals due to mixture or historical structure. To confirm the results, we use 3 methods: principal components analysis, kind chips, and structure. As you can see here, the principal components analysis divided the population in three groups: exotic lines, the small one, Mediterranean accessions, and northern types. And the three principal components explain 88% of the total variance. Here I show you the same results, but using a 3D scatter plot in which we can clearly distinguish the 3 groups I mentioned before. And here I show you the amount of individuals that was included in the right side in each of the collections. northern types 159, Mediterranean 186, and exotic lines from Chinese, Japan, Peruvian, and Thailand were only 24. GAPIT also display a heat map showing the differences between the accessions and also distinguishing the genetic difference between the groups, as you can see here. And finally, the Faba structure based on a variational Bayesian algorithm also distinguish the three groups that I mentioned before: exotic lines in red, the green Mediterranean accessions, and the northern types in blue. Then we started to analyze which statistical models was going to provide the best results in order to assign truly association between markers and traits. So we first analyze one trait, the plot yield, using the multilocus mixed model as implemented in Progeno, but we neither the Manhattan and cuckoo plots identify any significant SNP markers associated. The same happens when we use the Manhattan, the farm CPU method implemented in GAPIT. No significant SNPs were associated. But later on, we used the multilocus mixed model using the implemented in the R package, and we were able to identify 25 SNPs associated that explain nearly 84% of the phenotypic variance with a bone interest group of 5.7. Once we decide, we see that this R package provided good results, we started to analyze the same, use the same statistical model for other yield-rated traits such as pods per plant, seeds per plant, seeds per pod, and 100 seed weight. As you can see, in all the cases, we identify significant QTS for these traits. We also have started to perform some genomic selection studies that, as has been mentioned before, use large amount of sets to predict the breeding value in order to choose the best lines in breeding programs. So the prediction model is based on the genotypic and phenotypic data from the training population in order to estimate the genotypic estimated breeding value. And we tested two methods, the GWAS and the QGWAS. The first time we used the genome-wide markers, and in the second case, only the markers identified with the GWAS analysis. As you can see here, for the four traits I mentioned before, for all the QTLs that were detected, the QGWAS gave better prediction than the GWAS. And also as happens before, as Hilde mentioned with soybean, a decrease in the size of the training population tended also to decrease the quality of the genomic prediction. So indicating that the fixed effects using this can aid to the prediction. So we are using this workflow to the rest of agronomic, adaptive, and qualitative quality traits. And all the significant SNPs identified with the GWAS are being validated with, if possible, in QTL mapping studies. So my conclusion and future prospects after the experience of the EUCLEG is that the GWAS that have been obtained with the EUCLEG is being a driver for the gene discovery in Faba. Once the GWAS output pass the statistical criteria and the validation, you know that the next step is the candidate gene identification by defining the physical interval. This is not possible in Faba right now because, as I mentioned before, we don't have physical map, but a reference genome assembly effort is underway, initiated by the NORFA consortium. Also, a pan-genome is underway, launched by the University of Helsinki and Luk in Finland. Several consensus map using several parental populations is also underway, and also using the annotated reference Faba transcriptome that has been recently reported. All these are very, very excellent genomic tools that will aid the upcoming genome assembly and boost the advance of the Faba genomics and breeding in the near future. And well, I think this is all. I just would like to acknowledge my groups here at the Faba, on the left, the group that was involved in the multiplications and DNA extractions and GWAS analysis, and the group that has been involved in the evaluation of the quality traits. And especially thanks to Natalia, that was mostly involved in the GWAS, and the guide and excellent support of Marie Begard. And also two colleagues, Ignacio Solis from Agrovegetal in Spain and Dejan Sokolovic in Serbia, that have provided excellent field evaluation data that have been used for the GWAS analysis. And that's all. Thank you very much for your attention, and I'm ready for the questions.
Thank you very much, Anna, for an excellent presentation, outlining with an excellent background to before EUCLEG and also the results that have been obtained. Are there any questions? None have been added to the on screen yet. I don't know if there are any questions that anybody has. No, I have a question, Anna. You mentioned that you were using the BLUPs for doing the mapping. Have you for the GWAS analysis? Have you looked at the results from individual trials to see how whether you get associations when you look them for things like yield?
For individual trials?
Yes.
Yeah, we have to look for individual trials. Yeah.
And do you get very different results from the different trials?
Yeah, there is a big difference between the Serbian and the Spanish results, because the Spanish results are much more homogeneous 2 years, and compared with the Serbian, the production of the seeds was not as good, and I think was a bad year with a lot of diseases and drought. So the yield production was very bad compared with the civilian results. So I think we are going to make some analysis separately in order not to include too much noise in the analysis.
So some questions have now arisen. The first one is, how did you deal with heterogeneity in the varieties whilst genotyping, as these are partially outcrossing species?
Yeah, well, we first tried to use to select the 400 lines from germplasm banks that we know that they were mostly inbreeding lines. So we tried to use the most homogeneous and homozygous lines as possible. So even we know that there is very heterogeneous florina and is partial allogamous. So we performed 2 years of multiplication in insect-proof cages in order to try to gain as much as homozygosity as possible. So nevertheless, as I mentioned in the previous slides, there was some percentage of heterozygosity, but we tried to keep it as less as possible.
Okay, thank you. And the next question is, what is the current status of the sequencing and construction of the reference genome for faba bean? You mentioned it briefly in one of your slides, but
yeah, because we are involved in the also in this initiative, in this genomic initiative, and they have already started to well, it's quite advanced. I think maybe next year could be some preliminary results on this topic. Stig Andersen from the University of Aarhus in Denmark is leading part of this initiative, and they are quite advanced.
And a further question: how has the multiplication of faba beans been conducted, and the following field trials? Did you need to cover with nets to avoid cross-pollination?
No, well, once the people take the data, take it in the field, but we didn't use the insect-proof cages because it's impossible. There were huge extensions of field to perform these field assays. So we only perform the multiplications in insect phages, and then the material they collected from the fields were not used anymore. I mean, this material was not used. The seeds were not used for further analysis. So at the beginning of the project, we tried to have all the seeds that were necessary for the 2 years of evaluation. And this material, yes, was multiplied and did insect multiplications.
Cool.
I don't know if I explained myself.
Excellent. So you didn't use seed harvested from 1 year for the next year's trial.
No.
Multiplied
this
it all
is a
at the
mess.
beginning. Yeah,
This
excellent.
is a mess. Only we keep on the insect cages the seeds that we wanted to use for protein analysis. You know, because we wanted to have the same environment seeds, but they were this a small set of seeds were used only for the protein analysis. But the rest of phenological traits were scored in open fields without any insect-proof cages.
Okay, thank you very much. I think that is the end of the questions for today. However,
Okay.
it is possible to contact any of the speakers separately by email if you have any further questions to ask. There will also be an opportunity tomorrow to post more questions. So I think now it only remains for me to thank all the speakers from this morning's sessions for the very illuminating talks, and I'm excited to hear more tomorrow, which will cover not only the other species groups, but also talks on development of genotyping tools for use in GWAS analysis, and also a talk on accessing genetic variation resources from gene banks and the best way to deal with that. So we look forward very much for tomorrow. And again, I'd like to thank everybody who's contributed today, and look forward to seeing you tomorrow. Thank you.
Bye.
Okay. So